Good morning, children. Welcome again in biology class. Today again we'll continue chapter genes and chromosomes, and the topic is crossing over. So crossing over means the mutual exchange of blocks of homologous genes between a pair of homologous chromosomes. It allows the recombination during meiosis. So how this process of crossing over takes place in an animal or a non-organism? So it involves the breaking and rejoining of chromosomes in a synaptonymal complex. During the zygotine and phagotine stages of prophase 1 of meiosis, pairing takes place in that way that similar parts of chromosomes they lie side by side by mutual attraction between them. The chromosome spreads longitudinally in diprotein stage into two chromatids, which are bivalent and now composed of 4 chromatids. The chromatids, which are originating from the same chromosomes, they are known as sister chromatids. The two non-sister chromatids during this process they come in contact at certain points, and it is the reason where crossing over takes place by breakage and reciprocal fusion of two non-sister chromatids. The points or place where homologous held together and exchange bits of chromatids are known as chiasmata. And the number of chiasmata in a chromosome it depends on its length. So here children see crossing over. So this is a nuclear envelope, and here the chromosome they are shortened and they become visible at the single structure. See here, and this is the centromere. In this pairing of homologous takes place. Now see, it forms a bivalent form. Then pairing of sister chromatids is here. Bivalent, it forms a single chiasmata here. Okay. Now again bivalent, it forms double chiasmata. Now these homologous chromosomes, they get separated and they are repelled to each other partially. Now these chromatids of homologous chromosomes, they continue to repel each other, okay, and bivalents assume particular shapes depending on the number of chiasmata. So crossing over, it is a highly precise process as two chromatids they exchange exactly equivalent segments, and except in very rare instances, then neither lost nor gain any gene. At each chiasmata, the two non-sister chromatids they exchange their sections so that the chromosomes carried in the gametes are new as they carry genes which were originally not located in them. So let's see the autosome and the sex chromosomes. In organisms where the two sexes are separate, certain chromosomes, either 1 or 2, in a diploid cell, they differ from rest in their behavior and staining reaction during cell division. Now these chromosomes, they determine the sex of an individual and are called sex chromosomes. And the rest of the chromosomes, they are said to be autosomes. In a diploid individual, there are 2n-2 autosomes and 2 sex chromosomes. In certain insects like grasshoppers and roundworms females, they have 2 sex or X chromosomes, while males have only 1, and thus females are designated XX and males XO. In humans and other mammals, most insects and many plants, whereas females have 2X chromosomes, whereas males have 1X and 1Y chromosome. Y chromosome, it is different in shape and size, and it pairs with X during meiosis. Thus females are XX and males are XY. Since male produces 2 types of gametes, X or O, in XO-type and XOY in XY type, they are said to be heterogametic. Females are homogametic. They are producing only one type of gamete with an X chromosome. So here is a Drosophila melanogaster. Female is larger in size as compared to the male. In birds and some reptiles, females are heterogametic while males are homogametic. So to avoid confusion with the male heterogametic, XY system is referred to WS system with males WW and females WZ. So how to determine the sex? Now sex determination deals with the study of factors which are responsible for making an individual male, female or hermaphrodite. In 1891, Henking traced a specific nuclear structure through spermatogenesis in some insects, and he observed that 50% of the sperm received this structure after spermatogenesis. Henking called this structure as X-body, but he unable to explain its significance. Later it was found that X-body was in fact a chromosome, that is why it was given the name X chromosome. So due to involvement of X chromosome in sex determination, it was designated as sex chromosome and the rest of the chromosomes was named as autosomes. Now human genetic disorders. Now the disease which are transferred from generation to generation are called genetic disorders or genetically transmitted diseases. They are congenital, means abnormal conditions are present at birth. These disorders, they may be due to incompatible genes, defective genes or abnormalities in the structure or number of chromosomes. They may be inherited or they may arise due to mutations. So, Mendelian disorders in humans. Most common prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. Mendelian disorders, they may be dominant or recessive and they may be linked to both autosomes and sex chromosomes. So let's see hemophilia. It is a sex-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny. In this disease, a protein involved in the clotting of blood is affected. As a result, bleeding does not stop, even a simple cut is made. The heterozygous female may transmit the disease to sons. The possibility of a female becoming hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic. Sickle cell anemia. Now, sickle cell anemia, it is an autosome linked recessive trait which can be transmitted from parents to the offspring when both the partners are carrier for the gene. It is controlled by a single pair of allele. Out of the 3 possible genotypes, only homozygous individuals show the disease phenotype. Now, the defect is due to the substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. The mutant hemoglobin molecule undergoes polymerization, causing the chain in the shape of the RBCs from concave to sickle cell structure. Phenylketonuria. It is an inborn error of metabolism which is inherited as an autosomal recessive trait. Now, the affected individual, it lacks an enzyme that converts the amino acid phenylalanine into triazine. As a result, phenylalanine, it is accumulated and converted into phenylpyruvic acid. It causes mental retardation. Now, thalassemia, it is also an autosome-linked recessive blood disease which is transmitted from parents to offspring when both the partners are unaffected carrier for the gene. Now, the RBCs are small, pale and short-lived. Thalassemia occurs primarily in populations from countries bordering the Mediterranean Sea. So here are the sickle cell. These are the sickle cell, normal cells, sickle cell. Now, it is an inherited abnormality in hemoglobin which causes the RBCs to break down too easily. Now, it affects both the sexual equally. Thalassemia differs from sickle cell anemia because in sickle cell, a quantitative problem of synthesizing too few globin molecules, while in thalassemia, it is qualitative problem of synthesizing an incorrectly functioning globin. So in thalassemia, absence or reduced synthesis of one of the globin chains results in the excess of the other. As a result, free globin chains which are insoluble accumulate inside the RBCs and form precipitates which damages the cell, causing lysis and resulting in anemia. So, albinism. So, it is a condition where the skin and the hair of the whole body appear colorless due to total or nearly total absence of melanin pigment. It is an autosomal recessive disorder against the normally pigmented skin and hair, and albino is always homozygous for the character. Absence of the enzyme melanocyte triosinase leads to the failure of melanin formation from tyrosine and the person develops albinism. Albinos, although led a normal life, have a strong aversion against exposure to sun. Now the disorders which are carrying out through chromosomal abnormalities. The autosomal abnormalities, Down syndrome. Now this is the best example of autosomal abnormalities. Now the condition is caused by the abnormality in the baby's chromosome. Since babies are trisomic for chromosomes 21, there are 47 chromosomes instead of normal 46. So it is also known as triplo 21. Now such individuals, they develop due to non-rejection of autosomes during gamete formation. When such a gamete is fertilized by a normal gamete of the opposite sex, a zygote is produced which is trisomic with respect to that autosome. Now, affected babies suffer from variable degree of mental retardation and heart defects, and usually recognizable at birth by a number of different physical characters. Now, such children, they are of a half happy disposition and have a great sense of humor. Now, sex chromosomal abnormalities. Now, non-rejection of sex chromosomes during gamete formation, which is followed by fertilization and development, it can lead to individuals with abnormal sex chromosomes complement. Klinefelter syndrome and Turner syndrome, they are the two main sex chromosomal abnormalities which are found in men. or the human beings. Now, Klinefelter syndrome, it is characterized by trisomy. There are 47 chromosomes instead of normal 46, and the individuals with this syndrome, they are phenotypically fairly normal, but they have very low sperm count and are therefore sterile in nature. Their genitals are underdeveloped and the body hairs are sparse. So most cases they have some degree of breast development and tend to have longer than normal legs and arms. Now Turner syndrome, it is characterized by monosomy type. The individual possesses 45 chromosomes instead of 46 and they are immature females. They are characterized by poorly developed ovaries, web-necked, broad chest with underdeveloped breast and below average intelligence. So another interesting sign which is present in the short for metacarpal and metatarsal bones. So this is all for today, children. In our next class, we'll learn for our next chapter. Till then, thank you and have a nice day.